This is kind of a cautionary tale about supplementation and captive rearing and captive breeding. And I'm going to talk about captive rearing and captive breeding sort of separately, but the two components are linked. So naturally, that's how I'm going to approach it. So here you have just a cartoon. It's basically salmon recovery and a bunch of salmon sitting around a table deciding what should be done. And typically in terms of recovery, we often think about captive breeding and captive rearing as opportunities or programs that are essential to these recovery, or supplementation to move fish to new areas, to have them establish spawning populations in new areas, etc. So I wanted to talk about the various cautions and things we need to think about when we start to implement some of these supplementation programs. So the first one I'm going to focus on is captive breeding. What happens when we captively breed fish in a hatchery facility? And one of the concerns is with genetic diversity, and the other is with altered sexual selection. So in essence, what we do when we captively breed them, we completely remove sexual selection, that component of what's going on. So this is a classic example of what happens when we supplement a population, and this is known as the Riemann-Laker effect. So the idea here is on the x-axis, we have the relative contribution of that supplemented population, those hatchery fish we add to a wild population, which in this case starts off at 200. And we either supplement it with 2 individuals all the way up to 100 captive individuals in that population. On the y-axis is what we call total effective population size. So it's a number of breeders that are in the population. If we reduce that amount, then we're reducing the genetic diversity in the populations. And what the figure shows is that when you initially supplement a small amount, yes, there's a boost to the population in terms of genetic diversity and supplementation. But as that contribution of those supplemented individuals begins to get greater and greater, the actual effective size of the population declines, and we have a loss of genetic diversity. So that's one of the concerns in terms of supplementation. If those supplemented fish represent such a large proportion of the population in the end, we're reducing the genetic diversity and the effective size of it. The other thing I wanted to talk about was altered sexual selection. There's all sorts of things that are happening when salmon breed. Of course, there's things like mate choice, females choosing males, males choosing females in which to mate with, and that may be for good genes. They actually know what they're looking for when they're out there. They may be looking for compatible genes, things that are compatible between the 2 individuals that will make their offspring highly successful and fit in the new environment when they come out. We also alter that male-male competition that's going on. There's quite a lot of competition that goes on. That figure here shows you some of the competition. This is with brown trout. Unfortunately, I don't have the same thing with salmon, but exactly the same thing happens. And we have alternative reproductive tactics. In captive breeding programs, we almost never use these alternative precocial males in the spawning populations that we create, those captive populations that we release into the wild. And of course, we don't let the female choose the nest site that she's going to create, and we don't have any selection on their capabilities of creating an effective nest site. So we've altered all this selection. The second part I wanted to focus on now, so we've talked about captive breeding, now I want to talk about the captive rearing effects. And of course, we bring the fish from the river, we bring them into the hatchery, we breed them, and we rear the young for various stages, various lengths of time. And what happens within that area, we start with eggs, we create the spawnings that occur in captive breeding sections. Those evolve and are developed, should say, in the captive environment. And there's all sorts of things that go on. There can be selection, there can be phenotypic plasticity in response to that new environment, creates a new phenotype in the fish. So I wanted to talk about 2 studies that we've done looking at some of those effects. And the first one is to look at environmental enrichment during this very early stages. How important is the environment in which the eggs develop and the offspring develop influence their phenotype subsequently? And then I want to ask, is there transgenerational effects? Some of the effects that we see in the first generation in the hatchery, does that transfer actually into subsequent generations? So I'm going to focus right here on the early life stages. We're going to look at the development of the eggs in the nest. So in a gravel nest compared to a hatchery environment, so we have a complex environment in nature in the gravel. We have a simple environment in a captive environment. So basically there's no gravel, they're sitting in a heath tray or some sort of environment such as that as they develop, and then they emerge and begin feeding. We're just going to look at that stage from when they hatch to when they emerge and begin feeding. And I'm going to ask a series of questions about shaping the phenotype. What do those fish look like if they come from a complex environment versus a simple environment? And is it going to affect their morphology, their behavior, neural development, the brain development, and whether there's differences in the expression of genes when they emerge and begin feeding? And ultimately, I want to tie that into the fitness consequences in terms of survival and growth for a period of time once they emerge and begin exogenous feeding. So the first set of experiments is shown here. We have one master's student, Jim Wakuski, who was doing this work, and we had complex environments shown down the bottom right, and a simple environment shown here. We have multiple replicates of this, and we rear the fish in those environments. We look at shapes and morphology. The fish that emerge from the complex environment are larger in terms of weight, they're in better condition than those from the simple environment. So in essence, they're heavier, in better condition. In terms of their behavior, these first feeding opportunities for these juveniles that come out, this is their just first feeding opportunity. They're presented with artemia. There's differences in their ability and likelihood of feeding at that stage. Those from the complex environment have greater feeding response than those from the simple environment. There's also differences in the response to predators. We expose those juveniles right when they're emerging to a predator, a simulated predator, and those that are emerging from a hide, they take shorter time. If they're raised in a simple environment, they take longer if they're coming out from a complex environment. So what this means is basically those that are coming out from a gravel incubated environment are having enhanced feeding and are more risk averse. They're more responsive potentially to predators. We found no difference in brain development. There's been studies that show that brain development can be influenced by this complex versus simple environment, but we found no evidence of that. And then we looked at the expression of genes. And so this is a heat map showing it's from a 44K microchip that looked at the expression of genes. And we found that the expression of genes, there was about 808 differences in genes that were expressed at that stage. Of those, we could actually classify 184 of those by ontological terms, and we saw that most of those were related to things like mitosis and cell cycle, muscle development, heart development, things that were quite important during growth. Those types of things tended to be upregulated in the fish coming from the simple environment, and it was suggested that that might be a response to stress that those coming from the simple environment were exposed to versus those coming from a gravel incubated environment. Then we asked whether all these differences translate into differences in fitness. And so we had a semi-natural stream shown here on the right. We introduced those fish, 40 from a simple, 40 from a complex environment. We had 8 replicates. This was done for 42 days, right from when they start feeding for the next 42 days, and looked at their performance. What we found is that those, not too surprising given all the other things that we saw, those coming from the complex environment had greater survival than those from the simple environment. They also grew better than those from the simple environment. So in essence, they appeared to have higher fitness, at least for those first 42 days, in a semi-natural environment. So a good indication that there's multiple biological scales, there's differences in impacts on the fish that are emerging from simple versus complex environments. The more natural the environment, the greater the behavior and complexity of phenotype that comes out. And we're surprisingly that a lot of these things can happen very early in the life stages of these fish. So that's an important take home from that. So what I wanted to focus on this last bit of my talk was, are there transgenerational effects? Do some of these effects that we're seeing in terms of captive rearing actually translate to the next generation, even though they're born and raised in the same environment? Do the effects of their parents have any effect on the offspring in the next generation? And we're going to use the captive breeding program that is in the Inner Bay of Fundy. And here basically what's happening is they're taking juveniles from the river, bringing them to the hatchery, raising them up to adulthood in the hatchery, and then they may release the adults, or in this case, we're going to look at the offspring of those adults that come from that environment and seeing how they perform. So in essence, we have 4 different lines that we're going to look at. And we base this on an established pedigree that exists in this captive breeding program in the Inner Bay of Fundy. So we have individuals that are fully captively bred. They went from egg all the way up to adult, that they were fully captively bred. We'll look at the offspring of those adults. We then have individuals that were wild exposed. They were bred in the hatchery as eggs and fry, released early on. They spent 1 year in the wild, captured, and then brought into the facility and raised to adult. We look at their offspring. We have wild exposed too. Basically, they have 2 years in the wild. And then the final group is wild fish, that they're offspring of wild fish spawning in nature, and then they're reared for 1 year in nature and then brought into captivity. And then we look at the offspring of those adults at that next stage. We have a whole series of families. We had anywhere from 8 to 11 different families for these different groups. And then once they began to feed, they were released into a wild environment. They basically sorted and released into an environment. And in the environment shown here in a place called Bunnell Brook, we had 2 sites where we released the fish. We released about 12,000 juveniles just beginning to start feed. And we looked at the performance in those 2 sites in Bunnell Brook. This is the Inner Bay of Fundy here. You can see up on the map there in Canada where it's located. We then went back in September, October. Electrofish captured the offspring. We actually captured about a little close, 368 and 341 from the 2 sites. We then used genetic pedigree, basically pedigree, to assign the individuals back to the various groups. And we could do that with an 87 to 97% success rate. When we look at their performance, and this is a key slide from this, and looking at the performance during those first period of about 4 months in the wild environment, we have the captive bred individuals. These are the offspring of fish that were raised all the way from egg to adult in the hatchery facility. We have 2 sites, which is a gray site 1, and site 2 is the open white bars. And we can see that as we move along, that the survivorship of the fish is greatest when they're being exposed for 2 full years to the wild before they're brought in, compared to these captive individuals. These are the offspring of captive individuals for their full life cycle. So clearly there's indications that there's some sort of transgenerational effect on the performance of the offspring. So we can see that basically the wild exposure increased by about 2-fold the survivorship relative to the captively reared individuals during those first 4 months. We also note that the wild one, those fish that are wild and were reared in the wild environment for 1 year, and those wild exposed, they were captively bred but wild exposed for 1 year, had intermediate performance, but they were equivalent in performance. When we looked at the overall size of the different groups, there was no difference in body size. So you might say, well, after 4 months, is there anything that happens after second summer? And in fact, there is. The patterns remain fairly consistent, though they're dampened relative to what we saw during the first 4 months. So basically, it remains relatively consistent. So the question then becomes sort of what underlies this? And there's a number of things that might underlie it. We don't know the answer to that. So one might be intense selection has gone on for heritable variation that affected performance of the individuals. There might be epigenetic effects, things that have happened in the hatchery that affect the expression of genes when the offspring are released in the wild, transferred from parent to offspring. There may be maternal effects, differences in, for instance, egg size between the different captive groups. We didn't have the information on that, unfortunately. So just to wrap up with the conclusions from this, basically, we can see that captive breeding itself will alter selection. It affects genetic diversity, removes sexual selection. We can see that salmon are highly responsive to their environment, even during this very short life history period. And this can have transgenerational effects. The effects that happen early can be transferred to the next generation by various means. So what does this say for us? Well, it basically says that we need to think about harnessing and understanding what's going on to be able to actually improve our restoration programs and how we can effectively implement them. and that captive breeding and rearing done in an appropriate way can be a valuable tool, but we need to recognize that it should only be a temporary tool. And finally, that it should not inhibit other restoration or recovery means, that if we're trying to restore populations, these are some of the critical things. What is the cause of the decline in populations that we're seeing? And this captive breeding and rearing programs will not be sufficient on themselves to restore the populations unless we address the other components. So, and I'd just like to thank the various funding agencies and the collaborators on this program. Thank you.